This experiment screens chemical libraries for compounds that modulate post-transcriptional control mechanisms via the 3' untranslated region of transcripts. First, identify the 3' untranslated region of interest in the targeted gene. Clone this 3' UTR into a luciferase control reporter plasmid to create a luciferase 3' UTR chimeric transcript. Then stably integrate the two plasmids into a disease-relevant cell line. Seed the cells carrying the chimeric transcript in 96 well plates, treat them with a library of compounds for 24 hours, and then identify the molecules that affect luciferase activity. Cherry-pick the selected molecules for further validation screening, and next, treat both the control cells and the cells carrying luciferase 3' UTR chimeric transcript with a cherry-picked library subset. Results will differentiate if the effect of a compound is mediated via the 3' untranslated region or via other vector sequences.
Unlike the most reporter-based screening assays that focus on transcriptional regulation, this reporter cell-based assay focuses on identification of compounds that modulate mRNA transcript levels via its 3' untranslated region.
Harvest the stably transfected neuroblastoma cells by trypsinization. Dilute the cells to a concentration of 2 × 10⁵ cells per milliliter in rPMI containing 10% FBS and 1% L-glutamine. For 27 plates, prepare at least 250 milliliters of cell suspension, taking into account liquid handling, trough dead volumes, and repeated pipetting steps.